Hello again. So I'm going to talk to you today about mutations and how we use them in the lab. So you guys have been doing a lab, and you're going to finish it today, I think, looking at kind of natural mutations that are just kind of part of your DNA. In the lab, there are times when we specifically want to make a specific mutation, and that's what you guys are going to do with me in this lab that we're going to start today. So I'm going to talk about why we might want to do that. I mentioned last time about this protein, green fluorescent protein. I was talking about why we use this. And so this is a jellyfish, and jellyfish naturally have this GFP gene in them. They glow green. So at some point in the past, probably about 30 years ago or so, people decided, hey, it might be kind of interesting if we could take this gene and put it into other organisms. So using PCR and using some other molecular biology techniques, people copied the gene out of jellyfish and started putting it into other things. So we now have a full array of GFP animals. So I showed you guys the mice last time, but we have, I think this is a bee or a wasp, we have GFP worms, we have GFP bacteria, we have GFP rabbits, there's GFP kitties, you can buy yourself a GFP fish, they're called blowfish. So we've taken this gene and we've put it into a bunch of other organisms. And in general, that might be kind of cool, but it's not, I don't know, there's not a ton of science that happens here by turning something bright green. However, we've also taken GFP, and instead of putting it into a full rabbit to make the full rabbit green, we can actually use it to look at specific parts of cells. So these are a much smaller scale. Now we're looking at individual cells, and we can use that GFP to look at different structures. If you guys are familiar with mitochondria, basically these little tubular things in green, those are mitochondria. So we can use GFP not just to make pretty things, but we can use it to look at things in cells and track things in cells and watch things in cells. And so I actually use GFP all the time in my real job. So I work with bacteria, so I would study cyanobacteria. And these little green dots that you see are actually DNA. They're chromosomes that I've added GFP onto. So the GFP is sticking on them. And what I do in my life is I make movies of cells as they grow, and we can watch these little green dots as they divide, as they separate into different cells. And so this is kind of what I spend my time working on. So GFP is really, 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 really important for us to be able to use in kind of a lot of the biology that is done currently. Here.
Kristen, you said that it's expensive, like the reagents are expensive. On what level of expense are we talking?
So I cheaped out on you guys, and I split a 10 reaction kit into 16 reactions. That kit was $500. So the things that, the types of biology to do the things that we want to do, it's expensive. Especially if you want to be really accurate with what you're doing. So if you're just doing a PCR reaction, you can probably do that for, like, I don't know, you could do 100 of them for $200. You maybe do $2 a reaction. But if you really care about the sequence of your DNA, which we are going to talk about why we might care about that, and you guys might care about that for your previous lab, it's going to be a lot more expensive. So we talked about GFP. But sometimes you might want to look at more than one thing in a cell, right? There's not just mitochondria in a cell. There's mitochondria, there's endoplasmic reticulum, there's a nucleus. This is another example from my work. So these kind of white outlines, this is a single bacterial cell. And what you can see is that I have two different types of structures here. I'm not going to tell you what they are. But they are different. But I couldn't really tell that if I didn't have different colors. So we use not only GFP, but we use different colors of fluorescent proteins, and then we can combine them to get more information out of the systems that we're studying. And in order to do that, we need more colors. So there's been a lot of work put into taking fluorescent proteins that we know and love and making them into different colors. And the ones you guys are going to make today, you guys are going to go from GFP into yellow fluorescent protein, YFP, and blue fluorescent protein, BFP, and cyan, a fluorescent protein, CFP. So all of these things actually came from GFP, and people have been working to make them into this broader spectrum of colors. And there's some other colors that actually didn't come from GFP that we're not really going to talk about too much. But in general, where we're going here, GFP, green fluorescent, to YFP, which actually looks a little bit more yellow than this, CFP, cyan, BFP, blue fluorescent protein. So each group is only going to make one of these mutations. You're not going to make all mutations. But also, just so you get a sense of where we're at as a community, we have made fluorescent proteins that kind of span the gamut of colors. So you can have a rainbow of fluorescent proteins in your cells and look at a whole bunch of different things all at the same time. But these ones are the ones that we're going to look at right now, the GFP derived. So,
strawberry, cherry.
Yep, they named them. They started naming them after fruits at some point. So they started off just with the normal colors, and then somewhere we went into plum and grape and whatever.
I mean, hey, they are different colors.
They are different colors. They are slightly different colors, and if you have the right microscope and the right filters, you can actually tell the difference between M grape 1 and M cherry. They are different enough that you could actually tag mitochondria with one of those, and you could tag the nucleus with the other one, and you could actually see a difference. So very, very powerful tools for us, but actually kind of not that hard to change. And that's what we're going to be doing today. So you guys are going to be using PCR, like I talked about last time, to start making mutations. Yeah?
The right side says RFP1 derived. What is RFP1 from?
What is, if YFP is yellow and BFP is blue, what is RFP?
Well, like the GFP,
Red fluorescent protein.
the GFP is from the jellyfish. Is the RFP just from the GFP?
No, the RFP is, I think, from sponges. Like a sea sponge, yeah. So a lot of the colors that we end up getting, we get from more exotic types of animals because those are the ones that naturally fluoresce. So people have kind of gone in. I think RFP was actually two proteins that when they come together make RFP, and they've now found a way to combine those into a single protein and then mutate that into all these other colors. I looked into doing that, that was harder. So we're doing the easy one.
Awesome.
Yeah. So, how do we do this? As it turns out, it's not that hard. So this is the DNA sequence for GFP. Now remember, DNA is double-stranded. I'm only showing you the sequence that actually gets turned into a protein. And these colored nucleotides, these are the ones that need to be changed to make BFP, CFP, and YFP. So you don't have to mutate this protein a lot. These are just single amino acid differences when you actually change them. But we do need to be able to make that mutation. And because these are very easy mutations to make, we can use PCR to do that. So you guys remember PCR from last time in your last class, lots of PCR, right? So we're going to do PCR, but it's going to be a little bit different in that we're going to use, same thing, 2 primers, but we're going to use a circle of DNA, which we call a plasmid. So the reason why this works, we're still going to copy, but this one primer has a mutation in it. So most of it lines up along the DNA, it matches up. But it's got this one spot in the middle that's going to be different. So when this primer sits down, it's still going to bind, and then it's going to copy the DNA that way. My other primer is going to sit down on the other side of the DNA and copy that way. So in a PCR, how do we get copies? We're going in different directions. Because it's a circle. So if we start this way, we're going to copy around that. And if we start this way, we're going to copy the other way. And then over time, we now have 2 brand new pieces of DNA that are totally complementary to each other. They'll bind together, except for this one piece right here in the middle that has a mutation. And after we do the PCR and copy and copy and copy and copy it, you're going to have billions of copies of your plasmid with that one single or 3 or 2 nucleotide changes in them. So now you've used PCR to copy almost an entire piece of DNA with a couple little mutations. Yeah.
Does every plasmid have the mutation, or is it the same thing where some don't, but most do?
It's a perfect question. So I showed you basically, these are the new copies. you have a PCR tube, you had to add a DNA template to it to start with. That DNA template doesn't go anywhere. However, we're going to talk about a way to get rid of that next time. So today, you guys are just going to be doing the PCR to make a bunch of copies with a mutant, and we'll talk about how to get rid of the original stuff later. Any questions on that? Yeah.
So for the activity we did over there with the paper clips, didn't we start with a plasmid? Right?
You did start with a
So, like,
plasmid.
why does this like just copy it in a circle, but the other one we had to like break it and then copy it in a line?
So, because of where your primers sit. So, this primer, remember, it's going to keep copying until it doesn't have anything else to stop copying it, right? And this primer is going to do the same thing. But because I want this whole thing copied, this whole circle is going to get copied. It's the same. Yours would have, if we'd kept it in a circle, you can just imagine it would have been a primer here and a primer here. It just makes it much harder to make extra little copies inside a circle. But it's the same idea. We're not going to break this apart like we did the last one. So you're not going to unwind it from a circle. It's going to stay in this circle. And we're actually going to talk a little bit next time, because we're copying one way and we're copying the other way, there is a little piece there that isn't totally connected. So I'm showing it as a circle. It's not quite a circle yet. Well, we'll get there next week.
In due time.
In due time, exactly. So yeah, so this is basically what we did last time, is we just had a primer here and a primer here, and we were only copying off a piece of that plasmid. This time we're going to copy the whole plasmid. It's just really hard to make that into a visual activity for anybody to do. So that's why you guys broke it. That kind of makes sense? All right. All right, so quick reminder, what do you need to copy DNA?
Primers.
Primers, you need primers. What else do you need?
Polymerase.
Polymerase, what else do you need?
DNA.
Nucleotides.
Nucleotides, and what kind of DNA?
Your DNA.
Well, not my DNA, because I don't have GFP in me.
Desired gene.
The desired gene, the template DNA, what you want to start with, and then you need a thermocycler to raise and lower that temperature over and over and over again. So let's go through these real quickly. Template DNA, this is going to be your GFP DNA, your plasmid. Primers, I'm going to give them to you. One's going to go one way, one's going to go another way. One has a mutation on it, the other one doesn't. DNTPs, I'm going to give you, and then DNA polymerase, that protein that actually does the copying.